Hello, everyone, and welcome to, let's say, not being biased, to my most favorite, let's say, lecture that I'll ever give to you guys this semester, Tumor Immunity. This is within my comfort zone. If you guys have any question, anything, let me know because this is something that I feel extremely, let's say, comfortable in answering questions. Okay, so tumor immunity. Let's start off with cancer. So I guess we've definitely all heard of this. This is basically the word that we shall not utter, almost like Voldemort, cancer. It's something that we wish never to actually heard, that we will ever hear a doctor tell us that we actually are diagnosed with. But sadly, I'm gonna be very extremely honest with you guys. If you literally look basically to the person, let's say in front of you or to your left or to your right, one of you guys is most probably going to get basically actually have cancer because the rate of cancer has been dramatically increasing over the past years. And sadly, it's actually not just seen in adults, but I'll give you an example, colorectal cancer. Colorectal cancer was a cancer that you mostly saw with people that were 55 and plus. Now there's a huge surge of people having colorectal cancer at the age of 40, very young age for cancer. So there's an issue between this. And of course, as we all know, cancer per se, cancer is a disease in where your normal cells are being transformed to be more malignant. So something is happening, and specifically down to its genetics, that there's a certain mutations that is causing the cell to proliferate uncontrollably. That's really, if you want to pinpoint what cancer per se is. And the issue with cancer per se is not that it's able to proliferate that into basically proliferate, is that it's able to metastasize. So let's imagine that basically someone has breast cancer. That breast cancer is not just localized to the breast tissue, it can metastasize to lymph nodes, but also to the brain and to other areas where are extremely sensitive, specifically your brain or your bones, where they can actually cause extreme pain and actually can cause a lot of death because you're going to sensitive areas where having these abnormal growths can cause a lot of disruption of a lot of homeostatic processes. So that's the issue with cancer per se. But thankfully, we have our immune system. The immune system was something that we used to always think was, a very complicated system within our body that was trying to, let's say, just protect us from foreign invaders like bacteria, viruses, fungi, parasites, you name it. But that's not true. Cancer, or sorry, the immune system has a huge, undeniable role in the protection of us, specifically when it comes to cancer. And this is actually a very interesting kind of fact that people have started to see. Within our body, we develop about 1,000 cancer cells a day. So imagine one day you have 1,000 cancer cells. Thankfully, they do not progress into a full tumor, so a whole body or a mass of tumor cells, which we call a tumor. They do not go into that because our immune system is able to sniff them, to detect them, and more importantly, to kill them off. So the immune system has a very important role in this. And honestly, we started to see because the immune system has an extreme important role in actually detecting and sniffing out cancer, people have then theorized about this thing of using therapies to boost our own immune system to kill cancer. Since it's so good at actually effectively killing them, why don't we create therapies to further boost them and further arm them even more stronger than they are right now in killing? How about that? That would be a great thing. Because think about this. Because we, for example, in vaccination, when we get vaccinated for, let's say, influenza, for COVID, or whatever basically virus or bacteria that we're being exposed to, what we're trying to do is we're trying to create an army of T cells and B cells that are able to recognize infected cells or microorganisms and kill them off. So we're trying to create an army inside our own immune system to do so. Well, why can't we do this also for cancer? Why can't we actually create a vaccine that's going to help our own immune system sniff out even better the cancer cells and kill them off? Because this would be great. You would have internally your own immune system helping you out to kill cancer, and more importantly, during the time of relapse. So let's imagine that your immune system is able to wipe off cancer, in many cases, people will relapse, will suddenly have a spontaneous, basically, regeneration of the cancer. Well, that doesn't matter because you have a T cell response, again, a memory response that is able to basically kill it off without you even getting any symptom. That would be the holy grail. And that's what basically the new current therapies in cancer, they're basically progressing. And I used to think that in Lebanon, this was basically not done, but I was wrong, thankfully. I started to see people here at AUB working for basically generation of new types of cancer therapies like CAR T cells, checkpoint inhibitors, which I'll be talking about to you guys in the later lecture. But so there's literally hope. And when I saw this, it really gave me hope that yes, we can finally be more up to date with our own cancer therapies. Because right now, the modern types of treating cancer are actually the use of cancer immunotherapies in combination with traditional therapies like chemotherapy and other targeted therapies that most probably you guys have learned before or will learn later. But cancer immunotherapy now has become a pillar of therapy. It's basically now almost used for almost every single type of cancer. So, what actually made us discover that our immune system is actually protecting us from cancer? Well, it all really started off from mice experiments, which is majority of the cases, I would say, from many discoveries, actually. It's actually mouse experiments. And we started to see that what we can technically do with mice, we can actually spontaneously induce mice to actually have cancer by giving them a lot of chemical carcinogens or radiation that will actually make them grow a tumor. Or also, we've shown that we can actually transplant a whole tumor to them. I do this almost on a daily basis, sadly. I do this quite often, actually, and where I actually transplant tumors to mice, and where I'm trying to actually learn a bit more about how the tumor interacts with the immune system. That's mostly my core research activities. But when it comes to basically the mice per se, we started to see that you can transplant tumors in mice. Sometimes they will grow and sometimes they won't. And of course, this has actually kind of made scientists think, why are certain mice are able to actually accept this tumor and others not? Well, it actually boiled down to the type of MHC. So if you took a tumor that has, let's say, MHCA, and you gave it to a mouse that has MHCA, it will work. But if you take this tumor that has MHCA and you give it to a mouse that is MHCB, it does not. Haha. That tells you that there is some sort of immune recognition of that tumor. And this is what they found out. So they did many different types of experiments, and one very famous experiment is what they did is they got this mouse, this yellow mouse. They injected it with this red tumor that was irradiated, which basically means that these tumor cells are not able to proliferate. They're dead. So they injected it. So this is technically a vaccine, if you want to put this in the most kind of simple way. We call this actually whole cell vaccines, which we'll learn in your last chapter what that means. So this mouse now has basically generated a T cell response against these tumor cells. You take the same mouse and you inject the same tumor, but alive, so viable cells. You see that this tumor does not grow because it actually has a T cell response against it. But if you inject a different, basically, tumor, so this is now your blue one, it grows. You have this nice bump of tumor cells. This was one of the first experiments that really told us, like, hey, there is a T cell development of T cell. So there's a T cell development against the tumor. So the immune system is able to recognize in specifically cancer, which is then something that really sort of help us to allow us like, okay, there is a very undeniable, let's say, role of our immune system in helping us kill cancer. And of course, they did many different types of experiments. And what they found out is that, for example, if we basically took this mouse and we removed its T cells, sorry, this one over here. So if we took this mouse, we removed its T cells, they would actually grow the red tumor despite them having basically been injected with it. So they started to see that mice that are deficient in T cells actually start to grow tumors. They fail to reject the tumors. They start to grow them. So they started to see like, okay, T cells are extremely important in rejecting tumors. They also found out that, of course, that you can actually take a T cells that are against a cancer, you can transfer them to a mice, and that mouse will be protected towards the cancer that basically the T cells are recognizing. So tumor immunity can be transferred by adoptive transfer of T cells from immune mice. So they started to see that T cells are actually a very important immune cell that are really helping us out in recognizing cancer. And specifically, what they started to see is that antigen presenting cells were taking tumor antigens, so antigens coming from the tumor, and creating a T cell response. Because these T cells that are now educated to kill the antigen coming from the tumor, they were basically going to the tumor cell and killing it. This could be an antigen presenting cell, but this also could be a tumor cell, because as we know, all nucleated cells have MHC1. So the tumor cell can actually present an antigen to a naive T cell to actually push this to be more activated and expand. So these antigens, we call them tumor rejection antigens as well, because they're really helping to reject the tumor. These hence tumor rejection antigens. And there's so many different types, which I'll be going into a bit more details in a bit. But really, if you want to see, let's say, the main kind of experiments that really kind of helped us understand the importance of T cells or the immune cells per se, how important our immune cells are in controlling cancer, it really comes really to Paul Ulrich. Paul Ulrich is basically a German scientist, a physician, that we dub him as the father of immunology. He's really well known in the field of immunology. He has, let's say, discovered a lot of key important things within cancer, within basically immunology. And one of them is that he basically proposed that the immune system actually monitors and eliminates cancer cells. He was one of the first people to actually say that in the 20th century. But it was later in the 1950s that Frank McFarlane and Lewis Thomas, they proposed basically an immune surveillance hypothesis. And in this hypothesis, what they said is that our immune system is constantly scanning the body for T cells, sorry, for abnormal cells, for tumor cells. And they basically were eliminating any cell that actually showed any sign of a basically cancer transformation. And then they basically put out this into stages. They called this basically elimination, equilibrium, and escape. So these three, which would make up basically this, let's say, kind of cancer immunoediting. So they dubbed this hypothesis of cancer immunoediting, meaning that the cancer was being edited by our immune system. And this basically consisted of three sequential phases. First, it was elimination, and then second is equilibrium, and third would be escape, in which I will be talking about them in a bit more details. So, elimination phase, it technically means that your immune cells are detecting the cancer and they're eliminating this. This can be coming from CD4 T cells, CD8, gamma delta T cells, NK cells, macrophages. This is a combination of adaptive and basically innate immune cells that are able to actually detect cancer cells and actually kill them off. So this is the elimination phase. And then you have the equilibrium phase. That during this killing, what happens is that your immune cells, sorry, your tumor cells are basically are becoming less, they're not growing, they're trying to become a bit different in a way they're not going to be able to be detected. So you can see here, basically, these are in the beginning, your tumor cells that are, let's say, very red. With this pressure of basically your immune cells killing them, genetically they will start to create a lot of instability. They will start producing a lot of different genes and will try to bit by bit become different in a way that our immune cells are not being able to detect them. And these become undetectable, and they basically are kind of in an equilibrium phase. They're not growing too much, and your immune system is just keeping them in check. They're not killing them, they're just leaving them in check. So this is called the equilibrium phase. Later on, basically, these now new tumor cells that are different will start to grow. And they basically have developed a mechanism to completely evade your immune system and they start to grow uncontrollably. And this is called the escape phase, where they actually start to escape and start to grow a proper tumor that your immune system is not able to control. So I'll go into more details into these. So the elimination phase. So the elimination phase is basically the concept in where your immune cells are able to detect them and kill them. These could be natural killer cells. Natural killer cells have a beautiful ability to detect any normal cell that has downregulated MHC. Where does this happen? Because remember, all nucleated cells have an MHC1, all of them. But cells that are infected with a virus, they downregulate. The virus is smart enough to bring down the MHC. This is because they're trying to be hide from T cells, because T cells will only recognize infected cells through MHC. So what viruses do, they downregulate the MHC. Well, cancer also does that, because remember, if you go back here, they're trying to downregulate this MHC over here. They're trying to bring it down. So they don't, this T cell is not able to actually take up the tumor cell. It's not able to recognize it. Well, okay, sure, you might have evaded the T cells, but NK cells will actually detect that. They will detect the lack of MHC and they will kill it. CD8 T cells, basically now the ones that are basically now, let's say, educated with a tumor rejection antigen, will be able to kill them. Macrophages also can be actually come and phagocytose them or release a lot of these kind of reactive oxygen and nitrogen intermediates, these ROS, that can kill them. You can also have dendritic cells coming up and actually causing this basically phagocytosis of the tumor cells and actually presenting tumor antigens towards the adaptive immunity to either the B cells or your T cells. You can also have other cytokines, specifically interferon gamma, which I'll be talking about later, or this type 1 interferon alpha and beta, which basically all stimulate actually antigen presentation. So you have a combination of adaptive and innate immune system, the immune system coming all together to eliminate them. And what usually happens, this actually happens quite often, is that your cancer cells, once they're actually, let's say, growing, something is going to make them actually die. Either they go into apoptosis or, for example, with chemotherapy. They will die and they will start releasing antigens from them. These antigens will be taken up by dendritic cells, which then will basically go into your lymph node and teach your T cells to start actually, being educated to actually recognize any cell that has these antigens, which is specifically your tumor cells. They start to basically, now that you have, let's say, an army of them, they will traffic into your tumor. They will recognize the antigens on these basically tumor cells and start killing them. And this is what we call the cancer killing wheel. It's this nice wheel and where basically our immune system is able to actually recognize it. This is actually, if you ever hear people that have basically have given chemotherapy, so you hear a lot of these cases where people that are given chemotherapy to basically try to kill off the cancer, and suddenly the tumor does not basically is not responsive to is responsive. It basically it dies, but not all of it, and it's still basically progressing. So what happens with these patients is that they're removed from chemotherapy and they undergo palliative care. Palliative care is that now you're not treating the disease anymore, you're just giving the patient a good lifestyle before they die. But suddenly, these patients, suddenly they have a full remission. Their tumor now is completely gone, despite not getting any chemotherapy or anything. Well, what we see with these patients is that during the chemotherapy, these antigens are being released and they induce this wheel. This immune response can take 2 to 3 or 4 weeks. So that's why it takes a bit of time, and then we start to see a T cell response. So if you ever hear patients given chemotherapy and basically they're not actually surviving, they're basically, let's say the chemotherapy is not really doing its job, but then suddenly, let's say a month later, do they basically have a spontaneous remission, and especially where they do not actually being given chemotherapy, it's mostly because of this generation of T cells against it. Okay, so now once basically the elimination phase actually goes in, you either enter this phase where basically the tumor cells are all actually killed. However, if this and basically sometimes the tumor cells goes into this equilibrium phase, in where tumor cells persist in a dormant or slowly growing state. They are basically not growing too much, and where your T cells are not recognizing them and they're just there. your immune system is continuously controlling the tumor expansion, meaning that it's killing them, but not all of them. They're basically letting them go back to, let's say, imagine you have, let's say, 20 basically tumor cells. They're killing 10. They're going to 10, and they're going back to 20, and the immune cells are killing them another 10. So they're basically in this equilibrium phase where there's not a lot of growth happening. However, during this time, this basically what's happening over here, you're creating basically a pressure. Because there's a pressure that the immune cells are basically being under attack in some sort of way, some immune cells will basically have a lot of mutation in their gene to, in some sort of way, kind of exit this phase and be able not to be killed by your tumor cell. This is where the tumor immunoediting actually happens because the tumor cells will start to acquire genetic changes which will allow them to actually reduce their antigen expression. So they're not going to be able to be actually targeted by T cells. They're going to be able to resist immune cell killing mechanisms by, for example, increasing a lot of proteins like CTLA4, PDL1. These are basically very important um, proteins that we all know that it can stop T cells. They are peripheral tolerance proteins that put T cells into anarchy, and they can also kind of have different basically production of certain cytokines that pushes this into immune suppression. So this is the whole immune editing. And of course, what happens in this basically immune equilibrium phase, you start to get tumor clones that have now new genetic basically abilities to evade the immune system. And this is called the escape phase. They acquire mutations that allow them to escape the immune system to avoid immune recognition. And basically, this could be, for example, the loss of tumor antigens, the reduced MHC class 1 expression, secretion of immunosuppressive cytokines, recruitment of regulatory immune cells, all different things that I'll be talking about a bit later on. But this is what we see, and this is where basically our tumor cells are able to evade the immune system and actually grow properly into a big tumor that will give us symptoms. So now I'm going to be talking about the experimental evidence that we see about immune surveillance, for example. So the experimental evidence that really kind of supported that there is actually something of a, let's say, kind of this kind of elimination phase was actually in mice. We saw that mice that actually didn't have perforin, this protein that NK and T cells use to kill basically target cells, if mice lacked perforin, they had a huge number of lymphomas. These are uh, basically cancer coming from your lymphoid cells, your B, your basically dendritic cells, your T cells, your NK cells. Mice also that lacked RAC proteins, these proteins that are very important for B and T cell generation, for the BCR and TCR receptors, also developed a lot of epithelial tumor cells more frequently. Mice deficient in STAT1 signaling, a very important signaling cascade, or let's say transcription factor that is important to induce basically inflammation. They started to see that they had actually susceptibility to tumor formation. So it really showed that if you actually have genetic mutations that made your adaptive or your innate immune system actually weak, you were actually much more susceptible to cancer formation. And we see this not just in mice, but we also see this actually in patients. We also started to see cytokines. We see a lot of cytokines have a very protective role in actually protecting us. An example are interferon gamma and this type 1 interferons, is the interferon alpha and beta. Because what these do is they increase a lot the antigen presentation, they increase MHC presentation, antigen presentation. Remember, there's ERA proteins, there's basically immunoproteasomes that are helping to cut these antigens basically to be presented in MHC1. But they also actually have a huge anti-cancer proliferation effect. That interferon gamma can actually go into your tumor cells and stop tumor cells from proliferating. Then basically we have this equilibrium phase, the clinical evidence, and during this equilibrium phase, remember that your immune system is kind of keeping the tumor cells in check. It's not fully killing them all. It's making them sure, putting them in check in this equilibrium phase and where they're not really growing and they're not fully dying. They're just there. And this really, a very important case actually demonstrated that this phase existed. So this was happening between between a kidney transplantation. So we had basically this donor that was giving basically a kidney to someone. Suddenly, we started to see that the donor developed melanoma. Interestingly, he had melanoma. But more interestingly was that the patient that took the kidney from this now patient that developed melanoma also developed melanoma. And people were baffled. I were like, but how? What's the coincidence that these two people now developed melanoma? And when they looked at the cancer cells that both developed, they were genetically identical. So they were the identical cancer cells that were actually given. They later found out that the donor actually had basically melanoma in this equilibrium phase. And melanoma is it loves to metastasize, and specifically to the kidneys. So what happened was that the melanoma went to the kidneys and they stayed there in this equilibrium phase. When they took this kidney and put it in this patient now received this kidney and it was given immunosuppressive drugs. So they were stopping these basically the immune system from keeping this equilibrium phase. So the melanoma cells were now basically being able to actually grow uncontrolled. This is actually was one of the clinical evidence of this equilibrium phase. Another thing that we see is people with immunosuppressed patients. These basically we see this a lot in patients that are receiving, let's say, transplants, and they're literally put in immunosuppressive drugs. You start to see that they actually have a susceptibility to certain cancers, not just to basically in this kind of case where they were given basically an organ that had basically cancer cells in them. And generally, these patients that are immunosuppressed, they tend to develop different types of cancers because again, the immune system has an important role in stopping cancer. But we see this also in this kind of disorder, which is called post-transplant lymphoproliferative disorder, PTLD. And what happens in this case is that these patients are infected with EBV, the Epstein-Barr virus. This is the mononucleosis virus, the one, the kissing disease. Basically, by 70 to 80% of people have this disease. It's very, very common. But what happens is basically Epstein-Barr viruses and infects B cells. And what it can do, it can actually cause B cells to transform into malignant cells. They can be produced into lymphomas and create the massive tumors. But what happens is that they are actually kept in this equilibrium phase thanks to your immune system. They're kept in check. What happens is people that are actually have some sort of immunosuppression, either through drugs or something else, like HIV, for example, they start to actually have these B cells that are infected with Epstein virus grow and transform into lymphomas. These tumor immune evasion, we started to see a lot of mechanisms. These are downregulation of antigen presentation molecules, the secretion of immunosuppressive molecules, a lot of different types of, let's say, mechanisms that I'm going to be talking about right now. So, as we said, either that the tumor suddenly sometimes what happens, it develops some genetic, let's say, mutations that suddenly it has no antigens. It doesn't have barely any basically antigens. So this could be no peptide, or they basically start to reduce the adhesion molecules that basically the T cell needs, or no costimulatory molecules. For example, they're basically the B7 or this kind of stuff. It doesn't have any of these kind of ones that push the T cell to kill. Or that suddenly this tumor starts to actually have mutations that makes their antigens resemble extremely, very similar to self-peptides. And this kind of induces actually tolerization. But again, you enter the peripheral tolerance. T cells are not going to really recognize a lot, self-proteins a lot. Other ways is that basically the immune cell will lose, for example, but basically this is the same as that the T cells may eliminate tumors expressing immunogenic antigens, but not tumors that have lost such antigens. So basically reducing the amount of basically antigens. However, this is more important. These ones is tumor-induced immunosuppression. The tumor cell per se can actually produce a lot of molecules that stop T cells from killing. This could be cytokines, like for example TGF-beta, interleukin-10, that basically they can do, they can suppress T cells from killing, or they can actually bring Tregs. So Tregs are coming in to stop anything. What the Tregs do is they stop any immune response. So they're bringing them. But what they can also do is produce PDL1, which basically acts like a basically kind of peripheral tolerance receptor. So they're stopping any T cell from doing any killing. They can also reduce other basically kind of enzymes like IDO, which also stop T cells. We'll talk about all these different ones in a bit. But what tumor cells can also do is they can express a lot of extracellular matrix, like specifically collagen, which produces a physical barrier that does not allow any immune cells to actually go inside. So it kind of produces this protective layer around them. So I'll be talking about them. So one, for example, molecule that tumor cells can express quite abundantly that stop immune cells from killing them is TGF beta. Is this transforming growth factor beta. TGF beta, what it does, it really increases the amount of Tregs. So Tregs start to come into the tumor. Tregs start to stop any CD8 T cells from killing. But what TGF beta does, it also stops any basically CD8 T cells, either T helper 1 cells as well, so CD4 T cells that are coming in to kill, but also stops macrophages. It stops M1 macrophages, these pro-inflammatory macrophages that are really there to basically eat. It stops NK cells from killing. It stops dendritic cells from actually doing any antigen presentation. It has a lot of immunosuppressive functions. It's there to stop any immune response. Another type basically that happens is myeloid-derived suppressor cells. So what can TGF beta do is they can actually also push the production of myeloid-derived suppressor cells. And what myeloid-derived suppressor cells are, these are cells coming from the myeloid lineage. These could be monocytes, these could be macrophages that have actually attained a suppressor phenotype. Thankfully, because of TGF beta, what they do is that they go to your neutrophils or to your monocytes or to your macrophages and they make them become more immunosuppressive. These MDSCs now they come in and they start to produce a lot of different basically cytokines that stop your immune system. They can also produce a lot of basically reactive oxygen species that impair immune responses. It can actually deplete essential nutrients needed for T cells. So it can actually remove a lot of very key important amino acids that CD8 T cells need to kill. They're there again to stop basically any immune response. So similar to your Tregs, they work very similar to your Tregs. So MDSCs, they're found a lot in, let's say, melanoma, ovarian cancer, lymphomas, but they're also found extremely in abundance with pancreatic cancer, with basically with colorectal cancer, with triple negative breast cancer, with kidney cancers. They are actually a huge problem with cancer, actually. And I'm basically developing therapies that are targeting these annoying cells. I'll be talking about that mostly on Thursday. Another thing is basically these checkpoints. So one checkpoint that we were talking about is this basically PDL1. So remember, basically with an antigen presenting cell, so you have basically this basically tumor cell over here. So the T cell has this MHC that presents its tumor basically antigen. Your T cell is able to recognize this, but what happens sometimes is the tumor cell expresses something called PDL1. PDL1 binds to PD1 on a T cell. This actually induces the T cells to go into anarchy. It stops the whole T cell from killing anything. This is something that you see a lot in the peripheral tolerance. So this PD1, PD-L1 axis. So what happens is that tumor cells start to express a lot of PD-L1 and it stops CD8 T cells actually from doing any killing. Because this PD-1 has this item motif. So remember this item motif is this basically inhibitory motif that stops the T cell. It pushes it into basically apoptosis or into anarchy. So there has been a lot of basically important antibodies that are trying to block this from happening. Another one that we know is basically CTLA4. What happens is basically tumor cells can produce a lot of TGF beta, and TGF beta increases CTLA4. And we know that CTLA4 also does a very similar kind of basically it gives this basically it really this has this peripheral tolerance kind of mechanism. So TGF beta is produced, it goes to your T cells and they upregulate CTLA4. and this can go into antigen presenting cells, which can stop any antigen processing happening from here, from your T cell to basically activate. It stops it completely. Another thing that basically can be actually released, for example, you see this a lot in glioblastoma cells. These are basically brain tumor cells. They produce something called IDO, this indoleadenine 2,3 deoxygenase, IDO, which is an enzyme that breaks down tryptophan. Tryptophan is extremely important amino acid that's needed for T cells to kill, specifically CD8 T cells. So what it does, it breaks it, and it breaks it into this kynurenine. And this kynurenine has this opposite effect of tryptophan. It actually goes to T cells and it stops basically T cells from proliferating. So tumor cells can produce this quite a lot and stop your T cells from doing this. And as I said, what also can happen is your cancer cells can release a lot of extracellular matrix proteins, specifically collagen, that create this physical barrier that does not allow any immune cells to actually reach your tumor. This is extremely actually important. A lot of new basically types of therapies are trying to block or let's say break this physical barrier over here. So these are basically all the different types of rejections, basically that you basically let's say mechanisms and where tumors are employing to evade your immune system. Now I want to shift into talking about more about these tumor rejection antigens because as we said, T cells are able to recognize tumors. And they're able to do that because T cells are able to only recognize antigens. So as I said, basically the tumors actually have these tumor rejection antigens, the antigens that are basically are helping your T cells to reject the tumor. These basically tumor rejection antigens have many different types. And the issue with some of these tumor rejection antigens, they can actually look very similar to your normal cells. Because again, cancer originates from a normal cell. It's a normal cell that has transformed into a cancer cell. So it actually has a lot of self proteins. And what happens is that even though you create a T cell response against your immune cell, against your tumor cells, sorry, you can also develop a T cell response against also normal cells. So you see this a lot with melanoma. So melanoma is skin cancer, and specifically you create a T cell response against melanoma. Great, it's killing your skin cancer cells, but it also can go and kill other basically skin cells, these other melanocytes, which are destroying, and you create something called vitiligo. Basically, when you have these kind of patches of white, of basically no melanin in there. So what are there are basically the types of tumor rejection antigens? They can come from different categories. These could be mutated proteins generated by point mutations. This occurs quite often in cancer, and these are called neoantigens. We'll talk about them basically in details. These can be cancer testis antigens. These are antigens that usually are very limited to basically your testes, it's coming from males, but for some reason, tumors start to express them at a high amount. This can be coming from literally your skin cells. This can come from your cervical cancer. This can come from ovarian. This can come from sarcomas. We don't know exactly what's happening there, but we see basically these antigens that are very only found in testes, specifically in male testes. Now they're found basically in tumors of different types. You have differentiation antigens. These are expressed by tumor and their normal tissue counterparts. These are, for example, let's imagine you have breast cancer, basically, or sorry, let's imagine that you have basically, for example, skin cancer. Skin cancer have very specific proteins like melanin. Melanin is only basically found in your skin. It's not found in your liver. It's not found in your kidney. It's only found in your skin. So basically, sometimes what happens with these basically skin cells that turn cancerous, they start to increase the huge amounts of melanin, which can be used actually as antigens. So these are called differentiation antigens. You also have overexpressed proteins. These are proteins that are expressed at a higher levels in tumor cells. We'll talk about them a bit later. These could be also abnormally modified proteins. These could basically, for example, have an altered glycosylation. These could also be basically proteins generated by abnormal RNA processing. Or this could be also viral oncogenes. Some cancers are actually produced by viruses. I'll again be going into more details about each one. So, as we know, what happens quite constantly within basically tumor cells is the tumor cells go through a lot of genetic instability, meaning that they will actually go through a lot of different mutations. And what happens? So let's imagine this is basically you have a finished protein, a protein that basically has no mutations, and this protein is very normal. And what happens, because it's self-peptide, our T cell is not able to actually recognize it. But during a mutated cell, specifically in cancer, there's going to be certain mutations, and where this protein now has a certain a point mutation, and where now when it's actually cut into small antigens, this is now a new antigen that basically our whole immune cells have not seen before. These are called new antigens, and these are perfect. These are the holy grail. This is what you want to see because these new antigens are extremely recognized by our T cells, and they do not cause symptoms of autoimmunity because these are mutated. You do not find them. These are not self proteins anymore. You do not find them in normal cells. So basically, these are let's say the same protein, but this has a point mutation that now gives you an antigen that is different. This is what you want. And we see this a lot with RAS proteins and P53 proteins. RAS protein is a very important protein. It's a very important signaling cascade protein that's very involved in the growth of cells. P53 is also a very important tumor suppressor cell. It's a protein that actually helps basically detect genetic mutations and can cause cells to go to apoptosis. Then you have your cancer test. These antigens, these are, for example, MAGE proteins or NYSO. These proteins, MAGE or NYSO, are mostly found in basically the testes of male. You do see this in females, but you see much more the mRNA levels. You never see this in the protein levels. So this is something that you only really see in male testes. But for some reason, certain tumors, because again, tumor cells have some sort of in genetic instability, they start their genome is completely scrambled that they start to express this MAGE and NYSA protein, NYSO. This could be, for example, melanoma. This could be kidney cancer. This could be sarcomas. This could be basically also other epithelial cancers like ovarian cancer or cervical cancers, and where normal tissue doesn't express it, but only tumor tissues will express it. And this can be extremely helpful because this is immunogenic. This T cells will recognize this, and you do not get a lot of basically autoimmune symptoms. Then you have differentiation antigens. These are genes that are expressed only in particular types of tissues. And you get this a lot with melanocytes. So you have basically antigens coming from tyrosinase, GP100, MART1, and GP75. These are all proteins that are all involved in this melanin pigment. You see these only found in melanoma. So what happens is that in skin cancer, where you have a huge abundance of skin cancer, they start to express abundantly tyrosinase, GP100, MART1, and GP75. These are only found in your skin. They're not found in your kidneys, they're not found in your liver, or whatever. They're just found in your skin. So let's imagine you have skin cancer. You can develop a vaccine against these, basically, these antigens because you will target the skin specifically. Of course, this is great because you're targeting your skin cancer, but this can also have effects because you're also targeting healthy skin. So there's basically pros and cons with differentiation antigens. Then you have overexpressed antigens. And I'll be using a very good example here. So basically here you have this HER2 receptor. HER2 receptor is a receptor that's actually very involved in T cell. Sorry, my bad. It's involved in more actually growth factor. It actually helps normal cells, epithelial cells specifically, to grow. They're found basically all over your cells, and they really help. They kind of are a receptor that pushes cells to grow. So what we see is specifically in breast cancer. This is your normal breast cancer. It has, let's say, 4 copies of HER2. In breast cancer, in specific cancers that we call them HER2 positive breast cancer, you see there's basically actually not 4 copies, but you have 10 or 20 different copies of HER2. And they're overexpressed, and they actually help the breast cancer to actually start growing like crazy. Because this is overexpressed, now we can actually use it as a target, as an antigen target, actually. So these are overexpressed antigens. These are proteins that are overexpressed, and these can be overexpressed in all different types. Difference between differentiation antigens and overexpressed antigens, both are overexpressed, but this is only conserved to a certain type of organ, while can be found basically in different organs. HER2 can also be found in cervical cancer, can also be found in ovarian cancer. So it's not conserved. It's not a conserved basically protein. It's just overexpressed. Then you have abnormal post-translational modifications. These are an example of this is basically mucin. So normal mucin, which basically is this nice glycoprotein, is heavily glycosylated. But sometimes in breast cancer and pancreatic cancer, this mucin is actually underglycosylated. And because it's underglycosylated, it actually now exposes a lot of antigens. Because what glycosylation does, it hides a lot these antigens here that are not reachable to T cells. Now suddenly these are reachable and can be presented. So this is basically one way, a new source of antigens. Another type is abnormal post-transcriptional modifications. These could be, for example, during your mRNA, when you have the splicing, you have your introns, your exons, and you're splicing to remove your introns just to get an exon, which then basically helps you to get an mRNA to create your proteins. Sometimes what happens in cancer is that introns are not spliced. They're actually now put together with exons in your mRNA to be transcribed into a new type of protein. So now you have a protein that is completely new because of now the introduction of introns. So you have an abnormal post-transcriptional modification happening. Then you have viral oncogenes. Viral oncogenes, technically what this basically means is basically these are coming more from cancers that are originating from viruses. So you see this a lot with papilloma, the human papilloma virus. It's a very, very contagious virus and very dangerous virus. So what papilloma does, it actually infects basically a lot of these different types of epithelial cells. Specifically, this can be mostly your cervical cells, or it can also bind to basically to your anal kind of cells that are in your basically anal mucosa, your anal epithelial cells. You also get them in your basically penis cells, actually, believe it or not, in your throat cells. They can infect all there. And what basically the human papilloma virus can do, it has these two proteins, this E7, and E7 inhibits this retinoblastoma. And retinoblastoma, what it does, it's a very important tumor suppressor gene or protein that it really helps basically your cells to go into apoptosis if it detects that it's actually becoming cancerous. So it actually inhibits it, and then basically has another protein called E6, the human papilloma, that binds also to P53, and it degrades it. P53 also has a very similar role as retinoblastoma, that it also helps your cells to basically go into apoptosis once it actually gets mutations that can be very actually cancerogenic, that can cause them to go into cancer. So because of this, you're inhibiting these two proteins. Now you're causing the cell to accumulate a lot of basically mutations. They will not be detected, and they will start to actually become very cancerous. And actually, cervical cancer, 99.9% of cervical cancers come from human papilloma. And what we see is because these cancers are produced purely because of papilloma, we have a source of antigens coming from the papilloma virus, specifically E6 and E7. So what we've done is we've actually developed a new vaccine called Gardasil, which actually targets the E6 and E7 antigens. So you have antigens coming from E6 and E7 proteins from the papilloma virus, and you're vaccinating them. And now you're able to actually kill your cervical cancer cells that actually have this E7 and E6. It only works if basically the cancer is originating from basically cancer. Sorry, if the cancer cell is originating from the virus is causing it. So this is basically all the different types of antigens here summarized all together. So thank you so much for listening. And if you have any questions, please let me know.